for this opportunity and for inviting me to come to this group. I'm going to use 30 seconds or 20 seconds just to give a plug to the American Medical Informatics Association, which has the world's largest annual informatics conference, and this year it's going to be here in Washington. It's in 3 weeks, starting November 13th at the
Jen, if you want, you can email that to Jen and post it on the meetup site.
Okay. So if you don't know about American Medical Informatics Association, you should check it out. They're about 3 blocks away, and if you join, mention me, I get a brownie point for Okay, so first I'm going to start off with a disclaimer or a non-disclaimer. The opinions expressed in this presentation do necessarily represent the official policy of the United States government. I love to say that. I was at Columbia University for 20 years. I've been at NIH now for 3 years. It's been a lot of fun. So I'm going to talk to you today very quickly about this project at NIH that I'm building. I'm the chief of the Laboratory for Informatics Development. The deputy chief is Elaine Ayers, who's sitting back there. So the entire Laboratory for Informatics Development is here tonight. And we are building, with the help of a lot of other people, a repository for clinical research data. So I have no bullet slides. I did a whole talk with no bullet slides. We have a hospital at the NIH, you may not know this, the clinical center, and we have patients at the hospital who are all subjects in research protocols. Their data goes into a health record, in our case, an electronic health record like they do at any hospital. But their data also go into institute systems. There are 27 institutes and centers, many of which do clinical research. And the individual laboratories have their own systems, and then there are what we call personal systems that aren't necessarily electronic, but the clinical research data, by which I mean lab test results, query, questionnaire results, anything that we're measuring on our research subjects, go into all these sources. Now the clinicians taking care of these subjects are doing so the way clinicians normally do, one patient at a time. They're seeing a group of patients, but they're concentrating one patient at a time, chronological data. They're very much in the moment. Now the researchers, you've never seen a researcher before, this is what they look like. Researchers are looking at the data from a different perspective. Okay, they want to look across these data. They don't care what happened yesterday. They care what happened 5 days after they gave a drug, which might have been yesterday for one patient, might have been 5 years ago for another patient. So this is a screenshot of the electronic medical record system. It's a commercial system that we have. It looks at patients one at a time, chronological order. So instead we've created the Biomedical Translational Research Information System, or BTRIS, which is a way to give the researchers access to the data. This is a map of the NIH. So we're down here someplace, and there's the clinical center up there. And so you can see all these different buildings, and they all represent more or less different institutes and centers. We've been collecting data from these now for about 1.5 years. And 2 years now, I guess, we started really getting the data from the 2 clinical systems at the clinical center. The current system that's been there since 2004, and the old system where we have archive data from 1976 through 2004. We have NIAAA. I'm not going to use up my valuable time with the acronyms. NIAID. We've been collecting data from these systems, and now this year we're working on images, other sources around the clinical center, and the NCI, National Cancer Institute, which is small in this picture, but actually very huge. It's the biggest clinical group. This is our data model. I don't have time to go through all this. For those of you who like data models, it's actually a very simple thing. This is all stuff about the researchers and the trials and sort of a traditional entity relation model. And this is about errors in data loading. It keeps track of all the data that we're bringing in. But really, it's this, and this is our research entities dictionary, which is a controlled terminology. That's our research data. That's all the data in those 12 tables. And they're actually very simple. There's 3 observation tables, and each of those has an entity attribute value table that kind of holds the overflow of things that didn't fit the standard data model. But the data model of these 3 tables is all pretty much the same. And so it's really just one big table of observations with a bunch of these details. And then we have another one for events, and they're also very similar. The difference between an event and an observation is an observation has a result. So it could be a test result or an amount that was administered or something like that. And the events are things that we want that happen to the patient. So I order something, I perform a test, those are the events, and then the observations and the things come back. So the data model, very, very simple, which allows us to get everything in place. So I'm going to cut through a couple of these slides, and we post these on the site or something. You can stop the video and look at the screens. But I'll just stop on this one. This is a report that everybody has to do. It's the most mundane possible report. It says how many subjects in my trial. When I show this to the NIH researchers, they gasp. I mean, I'm not kidding. They go, oh, I can do this on your system? This takes me weeks to do, and I can do it with a click, 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 and there's my report. So it's really going to change the way people are getting at their data. Not going to go through, I mean, from an informatics standpoint, not that exciting. We've got all the data together in one place, and now we're producing reports that say, get me all the Chem 20s, or anything that is like a Chem 20, and put it in a report for me. We have a big terminology of 140,000 terms now representing the data that we collect from all these different systems, and they are organized into a big hierarchy, so clinicians don't have to know what are all the different alkaline phosphatases. They can go down and look at a tree structure and say, oh, get me everything in this class of alkaline phosphatase. Those classes don't exist in the individual systems. We had to create those and organize everything into that ontology. Again, just, lab reports. I got 50 slides here. I can't go through them all. But I thought, let me see what happens if I try to collect data from 1976 to the present on one patient. I'll get all their liver function tests. So we've got the data in there. We've got all these different sources from different places, different time periods. And I said, let me do a query, get me all the laboratory data for this person. And I was able to plot the liver function test from next January or June 1st, 1976, all the way to the end of 2009. There are very few places in the world where you can do this kind of graph and just pull everything together. And you've got all these different data sources. And so these aren't the same tests even. They're coded under different names and different codes, different laboratory systems. But our ontology allows us to pull this all together and put it in one place. The other thing, besides giving researchers access to the data in their active clinical trials, we also give them access to de-identified data to reuse those data for other purposes. So we had to automate a lot of things, and one of them is this certificate that they normally would have to go through, fill out a form, send it to some office, wait for the response. Now they can do their search, and when they're finally ready, they go, oh, I like these data, I want to get permission to use them. They fill out something and get their form filled out. I decided to do my own search of de-identified data because somebody said, we think the serum albumin levels are decreasing over the years. So I said, fine, get me all the serum albumin levels from 1999 through 2009, and let me plot them, the mean and the standard deviation. And sure enough, the normal ranges have been dropping for the last 10, 11 years, and it's not clear why, but at least I could go in, instead of doing a study and pulling clinical records and trying to figure this out, very quickly I got hundreds of thousands of laboratory points that I could just go in and say, oh, you know what, there's something going on here. Maybe I need to investigate what's going on here. So my next talk might be on solving the world's serum albumin shortage or something. I'm going to skip the terminology browser stuff, really isn't time. Just there's our map. We're now next year, this current fiscal year, we're working in ICHD and HGRI, and eventually we'll get everybody in there. So what? There's this cycle that goes from hypothesis generation all the way around to recruitment and accrual and data gathering and reporting publications, and we think that Beatrice is going to serve lots of purposes in that cycle. And so it remains to be seen. We just got started. We turned on a little over a year ago. We turned on the system and growing. There's our website. There's our URL for the folks that there's a lot of NIH people here, so they can actually click on staff only and get the details. But from outside, at least, there's an outward-facing page that can get more information. Thank you very much.